Now I'll be introducing the next chair. Her name is Nida Nahid. Nida Nahid is from Bhagalpur in the Indian state of Bihar. She is pursuing a bachelor's degree in radiology and imaging technology at the Mehwish Institute of Science in Patna. She completed her 10th grade at the Ikra Public School and her 12th grade at the AMU Girls School, both in Aligarh. And apart from that, she enjoys cooking and writing. Without further ado, please welcome Nida Nahid.
Good morning, everyone. It's my pleasure to introduce to you Mehwish Mirza, passionate and driven individual with a deep commitment to both medical and research. Currently, Mehwish is a dedicated researcher at Institute of Neuroimmune and Pharmacology, Seton Hall University, where her work focuses on fascinating intersection of neuroscience, immunology, and pharmacology. Her research aims to shed light on how the immune system and brain interact, which is crucial for the advancing medical knowledge in various areas. Mavish has also made significant contribution to the field of clinical research, working with prestigious institutions like Columbia University, Clara Mass Medical Center. She has co-authored Impact. She truly embodies the spirit of hard work and dedication. Please join me in welcoming Mavish Mirza. The stage is yours, ma'am.
Thank you so much, Nida, for the wonderful introduction. Hi, everyone. My name is Mehwish Mirza. I would first like to thank the American Society of Science, Engineering, and Technology for this wonderful opportunity to present my research work today. And of course, I would like to give a big thank you to my mentor and PI, Dr. Suli L. Cheng, who is currently my research mentor at the Institute of Neuroimmune Pharmacology, or INIP, at Seton Hall University in New Jersey. And so today I'll be presenting my current in silico research project on alcohol drinking modulation of glioma in people living with HIV. So to begin, just before I begin talking about gliomas, HIVs, and the role of alcohol in the modulation of both, I was hoping to first talk a little bit more about gliomas. And so, as I hope many of you have, read in the literature and just heard from everyday conversation, gliomas encompass a large variety of tumors that originate within the glial cell population present in the brain and spinal cord. And so these brain cancers are actually quite, unfortunately, quite deadly with immense, with large metastatic rates and unfortunately small survival rates. And so these gliomas range in terms of grade. There's grade 1 gliomas, grade 2 gliomas, up to grade 4 gliomas. And these grade 4 gliomas are also known as glioblastomas. And although the focus of my study is not on one specific grade of gliomas, I was hoping to give you a brief introduction in case you weren't familiar with these types of brain cancers. And so my focus is on all four types of gliomas, and these tumors are the most common primary brain tumors of the central nervous system. And so the second main player of the study is the HIV virus, which is also known as the human immunodeficiency virus, which is a single-stranded member of the lentivirus genus. And if it's left untreated, it eventually progresses to AIDS, otherwise known as the acquired immunodeficiency syndrome. And so the focus of my study is to analyze the interaction between gliomas, HIV, and alcohol drinking. And so my central hypothesis is that alcohol drinking may dissipate the impact of HIV's anti-glioma effect through a pathway known as the macrophage alternative activation signaling pathway, which is induced by an imbalance of what is known as colony stimulating factors, abbreviated as CSF. And so essentially what my hypothesis is that there's an imbalance in the ratio of two of these colony stimulating factors. So this imbalance of CSF2 to CSF1 is what regulates the imbalance or dysregulation rather, of the macrophage alternative activation signaling pathway when one drinks or engages in alcohol drinking. And so in the central hypothesis, I mentioned the anti-glioma effect. And so to kind of give a brief description of what this effect is, I pulled up this study, which was actually performed just quite recently, a few years ago, in Zimbabwe. And so essentially what they performed in this clinical research is an analysis of HIV positive, HIV negative patients, as well as glioma and glioma positive and glioma negative patients. And what they found in this study is that HIV positivity might actually protect against these brain cancers or gliomas with a 42% lower occurrence, as you can see here, in HIV positive patients compared to HIV negative patients. And so what this suggests is some sort of a protective role of HIV against these brain cancers. And so my first research question is, what is it that's part of the HIV virus that might facilitate this anti-glioma effect? And so we hypothesized in this study that the acute inflammation that occurs during the onset of HIV infection might actually play a role in explaining how HIV protects against these gliomas. And so we all know that HIV infection is a chronic infection in nature, but it actually has an acute infection stage, as we can see in this immunopathogenesis figure of the HIV-1 infection. And so what we can see in this figure is marked in black is the viral load of the HIV virus. And essentially what that is the amount of viral titer that's present within the human body. And so we can see an increase, a rapid increase throughout these six stages of the HIV-1 infection life cycle. And so we can see these stages marked as 1 through 6 up above. And what we can see actually is starting at the onset of stage 2 into the first roughly 5 days of stage 5, we see an increase of inflammatory cytokines. And so that kind of ties into what I was talking about with regards to the acute infection stage of this HIV virus. And so that might explain this inflammation, might explain how the virus protects against the glioma. And so a major player of this inflammation or this increase in these inflammatory cytokines is actually the macrophage cell, which I will get into a bit in a few minutes. And so as for the materials that were utilized in this in silico study, there were a whole list of softwares that were used, but I wanted to highlight three major ones, and those are the IPA software, which was used to generate a lot of these pathways that you will be seeing in the upcoming slides, and it was used to kind of simulate how HIV would look like and the pathways that it regulates within the typical human cell. And Omnibus database, as well as QKB or QIAGEN knowledge and base, were also utilized in this study, with the former being used to analyze RNA sequence datasets, including the GSC 47474 dataset, which I will actually get into right now. And this dataset, in fact, was actually created, or part of it was worked on by my research mentor, Dr. Chang, as well as a few other collaborators. And so this data set, the GSC 47474 data set, was designed to determine expression differences in various brain regions, such as the hippocampus, the prefrontal cortex, of what are known as F344 or HIV negative control rats, as well as HIV positive transgenic rats. And this was done by using an analysis of RNA sequence data. And so in this data set, we had two treatment groups, nicotine and saline. And of course, it was a comparative analysis. However, since the compound of interest in this study, in my study, was not nicotine, we focused solely on the saline the saline treatment groups, and we simply did a comparative analysis of HIV positive versus HIV negative groups treated with saline to further understand this anti-glioma effect, as well as alcohol's or ethanol's modulation of this anti-glioma effect. And to summarize, we found that from this data set that there was a decreased activation of a pathway that I mentioned earlier, the macrophage alternative activation signaling pathway, in the HIV positive groups, which did not occur in the HIV negative group. And so this pathway, the macrophage alternative activation signaling pathway, as its name suggests, polarizes macrophages towards the alternative phenotype. And the alternative phenotype is simply just the M2 phenotype of macrophages. And so there are two phenotypes that macrophages typically present as, two main ones, the M1 macrophage as well as the M2 macrophage. The M2 macrophage is an anti-inflammatory phenotype, and it engages in anti-inflammatory activity by releasing a whole host of anti-inflammatory cytokines such as IL-10, TGF-beta, and CCL1, and it's often involved in tissue repair and modeling. However, there's another type of these macrophages which engage in almost an opposite sort of activity, which is pro-inflammatory activity. And so it releases a whole host of pro-inflammatory cytokines such as TNF alpha and IL-1 alpha. And so essentially this pathway polarizes these macrophages towards this anti-inflammatory M2 macrophage, meaning that there's an increase in anti-inflammatory activity and a decrease of pro-inflammatory activity. And so this kind of ties into, so if we go slide back and look at what the findings reveal, this indicates that there's actually a polarization towards the M1 inflammatory phenotype upon decreased activation of this pathway in the HIV positive group. So that means that ultimately there is an increase in inflammation, which ties into what I was mentioning earlier about the inflammatory cytokines and how there's a brief acute rise of these cytokines during the HIV life cycle, which explains how the HIV virus facilitates the anti-glioma effect. And so we then performed an in silico analysis to kind of summarize the cytokines that play a role in this anti-glioma effect. And so to summarize it, we see a whole host of cytokines here, IL-6, IL-1, a lot of cytokines that I mentioned earlier briefly. But essentially, to summarize this network pathway, what we saw is that we observed an increase towards the pro-inflammatory M1 phenotype and a decrease of the anti-inflammatory phenotype. And so ultimately, this resulted in a decreased risk of glioma. And so this pathway is a good summary of everything I've explained up until now, which is that of the anti-glioma effect, where we see a decreased risk of glioma upon HIV infection. So now what makes our study, our in silico study, a bit different, and the first of its kind actually in the literature, is that we're actually studying the effect of alcohol drinking on the anti-glioma effect. And so as you may know, unfortunately, HIV positive patients are 2 to 3 times more likely than the typical non-HIV patient to engage in alcohol drinking behaviors. And so that is why we asked the question of, well, what is the role of ethanol on the anti-glioma effect? And so this is what we found. Again, just to summarize this pathway, we start off with ethanol. And what we essentially simulated in this software is what happens when an HIV positive patient engages in alcohol drinking behavior. And what we found is that there is a decrease towards the pro-inflammatory M1 phenotype. So there's a decrease of pro-inflammatory cytokines released by the M1 phenotype, as well as an increase towards the anti-inflammatory M2 phenotype. And overall, this leads to an increased risk of glioma. And so this is quite surprising because it shows that, in fact, alcohol drinking reverses all of the protective behaviors that the HIV virus protects against gliomas, meaning that it almost reverses the entire anti-glioma effect. And so our next question then was, okay, we've established that ethanol likely reverses the anti-glioma effect of HIV, but how does it do so? How does it reverse all of this? And so part of what plays a role in ethanol's modulation of this anti-glioma effect are what are known as colony stimulating factors, or CSFs for short. And so essentially what we found is that under ethanol treatment, in this in silico model, we noticed two things. Number one, an increased expression of CSF2, and number two, a decreased expression of CSF1. And altogether, this explains why there is an increased risk for gliomas, because typically, in the typical non-HIV cell, we see the opposite of this. In fact, we see a decreased CSF2 expression typically and an increased CSF1 expression. But it almost seems as if the alcohol, from what we found, flips this ratio of comparative of the concentrations of these two CSFs. And so we see an increased CSF2 expression and decreased CSF1 expression, which increases the risk for glioma. And so just to summarize, previous literature has shown that this flip of expression of the CSF2 and CSF1 markers does contribute to neurodegenerative conditions. This has been established in other studies, but our study is the first of its kind to highlight that this imbalance plays a role in ethanol's modulation of the anti-glioma effect. And so altogether, this kind of reaffirms our central hypothesis, which is that alcohol drinking may dissipate the impact of HIV's anti-glioma effect through the macrophage alternative activation signaling pathway, which is ultimately induced by this imbalance of CSF2 to CSF1. And so in the future, we plan to transition to an in vivo rat study to further continue our research on this topic. And so I would like to thank, first off, my mentor, Dr. Suli L. Chang, who is currently my research mentor and PI at the INIP. And I would also like to thank my fellow researchers at the INIP who have continued to support me throughout my months working at this lab at Seton Hall University in South Orange, New Jersey. And I would also like to give a big thank you to the ACID Conference for providing a platform for this presentation. Thank you so much.